So today's topic is Glycolysis cycle. Still yesterday we have discussed about glycolysis, pyruvate oxidation, citric acid cycle. Today's topic is Glycolysis cycle. This is a very interesting topic. The Glycolysis cycle is a variation of tricarboxylic acid cycle, which is also an anabolic pathway occurring in plants, bacteria, protists and fungi. In the Glycolysis cycle பாத்தீங்கன்னா vertebrates-ல நடக்காது. So what do you mean by vertebrates? Vertebrates are the organisms that have backbone. Vertebrates-ல இந்த cycle-க்கான enzymes இல்ல. Vertebrates are not having the enzymes for Glycolysis cycle, and this Glycolysis cycle occur in plants, bacteria, protists and fungi. That is, most of the lower organisms and lower plants will be carrying out Glycolysis cycle. Higher plants la nadakadu. Higher plants abdina, those plants which are capable of synthesizing their own food materials using photosynthesis. Photosynthesis அப்படிங்கிற process எந்தெந்த plants ஆலெல்லாம் carry out பண்ணிக்க முடியுமோ அந்த plants எல்லாம் Glycolysis cycle choose பண்ணாது. Those plants, those germinating seeds, இப்பதான் germinate ஆகுது, seedல இருந்து ஒரு plant germinate ஆகுதுன்னா அந்த மாதிரியான lower plants எல்லாம் தன்னுடைய food தானே prepare பண்ணிக்கிறதுக்காக choose பண்ற cycle தான் Glycolysis cycle. The Glycolysis cycle centers on the conversion of acetyl-CoA to succinate for the synthesis of carbohydrates. As you know well, photosynthesis நடந்தா what will be the end product? Do you know that photosynthesis carry out பண்ணீனா என்ன end products கிடைக்கும்? The plants will be synthesizing carbohydrates. Carbohydrates synthesize பண்ணோம் இல்லையா? So carbon dioxide plus water gives carbohydrates plus oxygen in the presence of sunlight. இதெல்லாம் நீங்க school daysல you would have read. Photosynthesis அப்படினாலே carbon dioxide வாட்டரும் sunlight ஓட presenceல combine ஆகி carbohydrates-ஐயும் oxygen waste byproduct ஆவும் release பண்ணும். And plants release பண்ற அந்த waste oxygen தான் we are inhaling. So எந்த அளவுக்கு நம்ம வந்து plantation அதிகப்படுத்துறோமோ அந்த அளவுக்கு நமக்கு oxygen கிடைக்கும் அப்படிங்கிறதுக்கான reason அதுதான். But ultimately என்ன product நமக்கு கிடைக்கும்? End product என்ன கிடைக்கும்? Carbohydrates. அதாவது sucrose and starch are the carbohydrates produced by plants. Higher plants ஆல இந்த carbohydrates easier synthesize பண்ண முடியும். But germinating seeds can't carry out photosynthesis because it will not be having the ability to carry out photosynthesis by capturing sunlight. Sunlight capture பண்ணி carbohydrates synthesize பண்ணக்கூடிய ability அதுக்கு இருக்காது. அந்த மாதிரியான plants என்ன பண்ணும்னா ரொம்ப சிம்பிளான கார்பன் காம்பவுண்ட்ஸை யூஸ் பண்ணிட்டு இந்த carbohydrates synthesize பண்ணிக்கும். அந்த மாதிரியான சிம்பிளான கார்பன் காம்பவுண்ட் தான் acetyl-CoA. acetyl-CoAங்கிற 2 carbon compound utilize பண்ணிட்டு succinate அப்படிங்கிற அந்த intermediate form பண்ணும், and this intermediate is utilized for the synthesis of carbohydrates. See the overall reaction of Glycolysis cycle. 2 molecules of acetyl-CoA plus NAD plus 2 molecules of water gives succinate plus 2 CoA plus NADH plus H plus. Here we are going to form one NADH and H plus, and acetyl-CoA molecules, 2 molecules are going to be utilized here for the formation of succinate, and the CoA from acetyl-CoA is released as the base compound. This cycle was discovered by R.W. Breedenbach and H. Beavers in 1961, and they have found that this particular cycle was carried out in an organelle called glyoxysomes. This particular cycle is not carried out in mitochondria. It is carried out in a specific organelle called as glyoxysomes. These glyoxysomes are not present in human beings, not present in animals. It is present in plants and in some lower animals. Lower animals it can be found. That is, lower organisms it can be found. And as I have said earlier, it is just a variation of citric acid cycle. Citric acid cycle எப்படி variate ஆகுது? Just you see the cycle. நாம ஏற்கனவே citric acid cycle தரோ பண்ணியாச்சு இல்லையா? இதுல என்ன சேஞ்சஸ் வருதுன்னு பாருங்க. You minutely watch. Acetyl CoA combines with or condenses with oxaloacetate to form citrate. With the help of the enzyme citrate synthase, citrate is formed, and this citrate is converted to cis-aconitate through dehydration and hydration steps with the help of the enzyme aconitase. And cis-aconitate is converted to isocitrate. This cis-aconitate is converted to isocitrate through the process of hydration. Through dehydration, cis-aconitate will be formed, and through hydration, isocitrate will be formed. The citrate is converted to isocitrate. And isocitrate will be, as usual, in citric acid cycle, be converted to alpha-ketoglutarate, alpha-ketoglutarate to succinyl-CoA to succinate, fumarate, malate, and then oxaloacetate is regenerated. This is the normal process of citric acid cycle. How the glyoxylate cycle is going to be deviated from this citric acid cycle? The important compound formed in citric acid cycle is isocitrate. This isocitrate is going to be lysed, is going to be broken down. This isocitrate is going to be broken down to form glyoxylate and succinate. Isocitrate, if isocitrate lyase enzyme use பண்ணிட்டு glyoxylate ஆவும் succinate ஆவும் cleaver என்ன enzyme lyase enzyme help பண்ண போகுது. Glyoxylate அப்படிங்கிற compound இங்க நமக்கு ஃபார்ம் ஆகிறதுனால this is called as glyoxylate cycle. And glyoxylate is a 2-carbon compound, and succinate is also formed as the byproduct. And form on a glyoxylate enna pannuna, it will be combining with another molecule of acetyl CoA to form malate. Glyoxylate combines with another molecule of acetyl CoA to form malate with the help of the enzyme malate synthase. So two enzymes are going to be participating in glyoxylate cycle. One is isocitrate lyase and other one is malate synthase. It is not synthetase, it is malate synthase. Okay, so isocitrate is cleaved into glyoxylate and succinate with the help of the enzyme isocitrate lyase, and the formed glyoxylate will be combining with one more acetyl CoA to form malate, and CoA is released. And the formed malate will be converted to oxaloacetate, and oxaloacetate again condenses with acetyl CoA to form citrate. Citrate is converted to isocitrate. Isocitrate is cleaved with the help of isocitrate lyase to form glyoxylate and succinate. And succinate will be as such entering into citric acid cycle, and glyoxylate combines with one molecule of acetyl CoA to form malate, and malate will be again converted to oxaloacetate. But where the carbohydrates are synthesized in glyoxylate cycle, that we'll be discussing in the next slide. And this is the variation of citric acid cycle. Here all other enzymes will be as such participating. எப்படி சிட்ரிக் ஆசிட் சைக்கிள்ல மற்ற என்சைம்ஸ் எல்லாம் பார்ட்டிசிபேட் பண்ணுவோம் அதே மாதிரி தான் பார்ட்டிசிபேட் பண்ண போகுது. பட் எக்ஸப்ட் என்னென்ன என்சைம்ஸ் எல்லாம் பார்ட்டிசிபேட் ஆகாது அப்படின்னு பாத்தீங்கன்னா ஐசோசிட்ரேட் டீஹைட்ரோஜனேஸ் தேவையில்லை, ஆல்பா கீட்டோ குளூட்டாரேட் டீஹைட்ரோஜனேஸ் காம்ப்ளெக்ஸ் இஸ் நாட் நீடட், and succinyl CoA synthetase is not needed. Other than that, all other enzymes will be participating in glyoxylate cycle. The two key enzymes of glyoxylate cycle are isocitrate lyase and malate synthetase. And put the question, what are the two key enzymes of glyoxylate cycle? Just answer me. What are the two key enzymes of glyoxylate cycle?
Isocitrate lyase, malate synthase.
Malate synthase. Very good. Isocitrate lyase and malate synthase. The next slide, we are seeing the same thing. Here also you can notice that acetyl CoA combines with oxaloacetate to form citrate, and citrate is converted to isocitrate. Isocitrate is lysed to form succinate and glyoxylate, and glyoxylate combines with another molecule of acetyl CoA to form malate, and malate is formed with the help of the enzyme malate synthase. The two enzymes which are put in the box, that is isocitrate lyase and malate synthase, are the key enzymes of glyoxylate cycle. Now we have synthesized malate. Malate is again oxidized to form oxaloacetate, and oxaloacetate will be again forming citrate. So this cycle will be continuing, and this is called as glyoxylate cycle. glyoxysomes are specialized peroxisomes found in plants. Especially we can find this glyoxysome in germinating seeds. In the seed eல்லாம் இது germinate ஆகுதோ, please mute your audio. And germinating seeds will be having this specialized organelle called glyoxysomes, and glyoxysomes possess the key enzymes of glyoxylate cycle, that is isocitrate lyase and malate synthase, and glyoxysomes are helpful in the conversion of acetyl-CoA to carbohydrates during germination. The glyoxysomes also contain the enzymes needed for the breakdown of fatty acids. Fatty acids breakdown பண்ற enzymes glyoxysomes இருக்கும். அதே நேரத்துல gluconeogenesis carry out பண்ணி sugars, அதாவது carbohydrates synthesize பண்ணிக்க வேண்டிய enzymes glyoxysomes இருக்கும். That is the difference. Glyoxysomes also possess the enzymes of fatty acid breakdown, fatty acid oxidation, and synthesis of glucose, otherwise called as gluconeogenesis. And in the germinating seeds நான் ஏற்கனவே சொன்ன மாதிரி glyoxysome ஒரு specific organelle இருக்கு, அதுதான் glycoxylate cycle carry out பண்ணும் நான் சொல்லிட்டேன். ஆனா higher plants மாறும்போது you can't find this glyoxysome. Whenever it acquires the ability to carry out photosynthesis, it will be losing that particular organelle, and glyoxysomes only, it is only found to be present in germinating seeds. Seeds வந்து germinate ஆகும்போது வரக்கூடிய ஒரு organelle தான் வந்து glyoxysomes. And this glyoxysome also participates in photorespiration and nitrogen metabolism. And what are the connections between glyoxylate cycle and citric acid cycle? You can see here, that is lipid body. This is lipid body. The lipid body, that is the seeds will be having lots amounts of fatty acids, lipids. Germinating seeds will be having more amount of lipids. நிறைய lipids வந்து அதுல இருக்கும். அது என்ன ஃபார்ம்ல இருக்கும்னா triacylglycerol அப்படிங்கிற ஃபார்ம்ல இருக்கும். And those triacylglycerols will be broken down to form fatty acids. Triacylglycerols lipolysis ங்கிற process மூலமா fatty acids break down ஆயிடும். Triacylglycerolங்கிறது ஒரு complex molecule. நம்ம ஏற்கனவே அதை பார்த்திருக்கோம். triglycerides ஃபார்ம்ல தான் நம்மளோட fatty acids எல்லாம் போயிட்டு ஸ்டோர் ஆகும். அதுவும் எந்த ஆர்கனல் அப்படிங்கறதையும் சொல்லியிருந்தேன். Do you remember that? For எந்த ஆர்கன் அப்படிங்கறத சொல்லியிருந்தேன். Which is the organ used for the storage of triglycerides in human beings?
Adipose tissue.
Very good, very good. Adipose tissue is the organ where the excess of fatty acids are stored in the form of triglycerides. இங்க germinating seedsல நிறைய lipids இருக்கும். இந்த lipidsல இருக்க triglycerides எல்லாம் fatty acids break down ஆன உடனே this fatty acid will be entering into glyoxysome. You can see in the picture, glyoxysome is present on the left hand side and the mitochondrion on the right hand side. So you can see the fatty acids are entering into the glyoxysome. As I have said earlier, the glyoxysome also possesses the enzymes for fatty acid oxidation. Fatty acid beta oxidation மூலமா நாம வந்து ஆக்சிடைஸ் பண்றோம் இல்லையா, அதே மாதிரி glyoxysome என்ன பண்ணும்னா fatty acids oxidize பண்ணி acetyl-CoA அப்படிங்கிற அந்த final compound convert பண்ணிடும். And this acetyl-CoA will be entering into glyoxylate cycle. Glyoxylate cycle as usual என்ன ஆகும், acetyl-CoA combines with oxaloacetate to form citrate, and citrate to isocitrate. Isocitrate is going to be lysed into glyoxylate and succinate, and the formed glyoxylate combines with another molecule of acetyl-CoA to form malate with the help of malate synthase, and malate is converted to oxaloacetate. So இங்க form ஆன அந்த succinate இருக்கு இல்லையா, this succinate is going to be transferred to mitochondria via cytosol. Cytosol வழியா இந்த glyoxysomeங்கிற ஆர்கனல்ல இருந்து மைட்டோகாண்ட்ரியாக்கு அந்த succinate transfer ஆயிடும். Succinate அங்க போனதுக்கு அப்புறமா it will be entering into citric acid cycle. As such, the citric acid cycle la mitochondria la enter ஆயிடும். Enter ஆகி it will be forming malate. Malate form பண்ணும். ஆனா இந்த malate நாம ஏற்கனவே gluconeogenesisல பார்த்திருக்கோம். Malate is permeable to mitochondrial membrane அப்படிங்கறத நம்ம பார்த்திருக்கோம். This malate will be coming out of the mitochondrial membrane, and this malate using malate dehydrogenase, cytosolic malate dehydrogenase, it will be oxidized to form oxaloacetate, and this oxaloacetate will be entering into gluconeogenesis, that is reversal of glycolysis pathway. It will be forming hexo sugars, and finally the hexo sugars are combined together to form sucrose, that is glucose and fructose combined together to form sucrose. So finally our lower plants or germinating seeds have synthesized their desired carbohydrates in the form of sucrose. So 4 different organelles are helpful in that. One is lipid body, other one is glyoxysome, other one is cytosol, and the last one is mitochondria. 4 different organelles are combinatively involved in the formation of carbohydrates. And repeating again, the germinating seeds will be having more amount of triglycerides. நிறைய lipids வந்து ஸ்டோர் ஆயிருக்கும் சீட்ஸ்ல, விதையில நிறைய ஸ்டோர் ஆயிருக்கும். அந்த விதை தான் இப்ப ஜெர்மினேட் ஆக போகுது. அப்ப அந்த விதையில இருக்கக்கூடிய நிறைய லிப்பிட்ஸ் என்ன ஆகும்னா ட்ரைகிளிசரைட் ஃபார்ம்ல இருக்கும். அது பிரேக் டவுன் ஆகி ஃபர்ஸ்ட் ஃபேட்டி ஆசிடா மாறிடும். அந்த ஃபேட்டி ஆசிட் எங்க வரும்னா கிளையாக்சிசோமுக்கு வந்து ஃபர்ஸ்ட் அது ஆக்சிடைஸ் ஆகும். ஆக்சிடைஸ் ஆகி அசிட்டைல் கோஏவா மாறும். அந்த அசிட்டைல் கோஏ ஆக்சலோசிட்டேட்டோட கம்பைன் ஆகி சிட்ரேட் ஆகும். சிட்ரேட் ஐசோசிட்ரேட் ஆகும் கன்வெர்ட் ஆயிடுச்சுன்னா, and then this isocitrate will be converted to glyoxylate and succinate, and the succinate which is formed in the glyoxysome will be entering into mitochondria, and glyoxylate form ஆயிருந்தது மறுபடியும் இன்னொரு molecule of acetyl CoA ஓட combine ஆகி malate form பண்ணும், and malate will be converted to oxaloacetate. As such the glyoxylate cycle will be continuing in glyoxysome, and a form ஆன succinate எங்க வந்துரும்னா mitochondriaக்கு வந்துரும். mitochondriaக்கு வந்த உடனே fumarate convert ஆகும், and then it will be converted to malate. Malate now it is ready to go out of the mitochondrial membrane, and it is oxidized to oxaloacetate in cytosol, and oxaloacetate அதுக்கப்புறம் எப்படி குளுக்கோஸா மாறும் அப்படிங்கிறது உங்களுக்கு ஞாபகம் இருக்கா? So oxaloacetate with the help of the enzyme phosphoenol pyruvate carboxykinase, and with the help of GTP, a high energy compound, it will be converted to phosphoenol pyruvate, and phosphoenol pyruvate to 2-phosphoglycerate, 3-phosphoglycerate, 1,3-bisphosphoglycerate, and glyceraldehyde 3-phosphate, and glyceraldehyde 3-phosphate to fructose 1,6-bisphosphate. இது வரைக்கும் எந்த ப்ராப்ளமும் இருக்காது, because all the enzymes catalyzing the reversal of glycolysis will be in the it is capable of catalyzing both in the forward and backward direction. Reversible nature உள்ள enzymes ஆல fructose 1,6-bisphosphate வரைக்கும் ஃபார்ம் ஆயிடும். And fructose 1,6-bisphosphate is hydrolyzed to fructose 6-phosphate with the help of the enzyme fructose 1,6-bisphosphatase, and fructose 6-phosphate is isomerized to form glucose 6-phosphate. And what is the enzyme needed for the formation of glucose from glucose 6-phosphate? Glucose 6-phosphate should be converted to glucose with the help of the enzyme dash. What is the name of the enzyme? Can anybody tell me? What is the name of the enzyme helpful for the conversion of glucose 6-phosphate to glucose?
Glucose 6-phosphatase.
Glucose 6-phosphatase. Good. Which tissues or which organ lacks glucose 6-phosphatase in human body?
Muscles.
Very good. Muscle is lacking glucose 6-phosphatase. And now we have come to the regulation part of glyoxylate cycle. So this slide is very interesting slide, and you have to watch it carefully. You have to understand one point is citric acid cycle is carried out for the provision of energy, that is NADH and FADH2 molecules are formed, and those reducing equivalents will be entering into electron transport chain for the formation of ATPs. But glyoxylate cycle occurs for the formation of glucose, as well as some amino acids and nucleotides can also be formed as a result of glyoxylate cycle. So glyoxylate cycle oda aim வந்து carbohydrates synthesize பண்ணனும். Citric acid cycle oda aim வந்து energy produce பண்ணனும். இதுதான் difference. So அப்படி இருக்கும்போது ரெண்டுமே simultaneously நடக்காது. So either glyoxylate cycle will occur, otherwise citric acid cycle will occur. So depending upon the demand of the plant cell, எனக்கு எனர்ஜி வேணுமா அப்படின்னு பார்த்துட்டு அதுக்கேற்ற மாதிரி சிட்ரிக் ஆசிட் சைக்கிளை ப்ரொசீட் பண்றதும், இல்ல கார்போஹைட்ரேட்ஸ் தான் சிந்தசைஸ் பண்ணணும் அப்படின்னா கிளையாக்சிலேட் சைக்கிள் கண்டினியூ ஆகும். ஸோ இப்போ இதுல ஏதாவது ஒண்ணுதான் நடக்கும், ஒண்ணு நடக்காது அப்படின்னும் போது how it is going to be regulated அப்படிங்கறத நம்ம இங்க டிஸ்கஸ் பண்ண போறோம். So acetyl-CoA is converted to isocitrate. The common product of both the cycles are isocitrate. அது உங்களால புரிஞ்சுக்க முடியும்னு நினைக்கிறேன். ஏன்னா ரெண்டு சைக்கிளுக்குமே இப்போ ஒரு காமனான இன்டர்மீடியட் ஃபார்ம் ஆயிருக்கு, தட் இஸ் ஐசோசிட்ரேட். And isocitrate, if at all it is used up by isocitrate dehydrogenase, it will be entering into citric acid cycle. Isocitrate dehydrogenase use பண்ணினா citric acid cycle நுழைஞ்சிரும். Isocitrate lyase use பண்ணினா அது glyoxylate cycle நுழைஞ்சிரும். அப்ப which enzyme is going to be active? Either isocitrate dehydrogenase or isocitrate lyase. Who is going to decide that? அப்படிங்கறத நம்ம ரெகுலேஷன்ல டிஸ்கஸ் பண்ண போறோம். சோ இப்போ ஒரு காமனான ப்ராடக்ட் நமக்கு ஃபார்ம் ஆயிருக்கு, தட் இஸ் ஐசோசிட்ரேட். இப்ப ஐசோசிட்ரேட் ஃபார் எக்ஸாம்பிள் இஃப் இட் வாண்ட்ஸ் டு என்டர் இன்டு சிட்ரிக் ஆசிட் சைக்கிள், அப்ப என்ன என்சைம் ஆக்டிவேட் ஆகணும்? ஐசோசிட்ரேட் டீஹைட்ரோஜனேஸ் ஆக்டிவேட் ஆகணும். அண்ட் ரெண்டு என்சைம்ஸ் வந்து நம்ம மேலாங்க we have expressed the role of two different regulatory enzymes. One is protein kinase, another one is phosphatase. Protein kinase நோடைய ஃபங்க்ஷன் என்ன? What is the role of protein kinase? Protein kinase is going to phosphorylate. Kinase வந்தாலே it is going to transfer phosphate group அப்படிங்கறத நான் சொல்லியிருந்தேன். Protein kinase என்ன பண்ணும் அப்படின்னா ஐசோசிட்ரேட் டீஹைட்ரோஜனேஸ் உடைய அமினோ ஆசிட் காம்போசிஷன்ல ஏதாவது ஒரு அமினோ ஆசிட்ல பாஸ்பேட் குரூப்பை அட்டாச் பண்ணுது. பாஸ்பேட் குரூப் அட்டாச் ஆயிருச்சுன்னா it becomes phosphorylated அர்த்தம். Phosphorylated எப்படி குளுக்கோஸ் 6 பாஸ்பேட்டா கன்வெர்ட் ஆச்சோ அதே மாதிரி ஐசோசிட்ரேட் டீஹைட்ரோஜனேஸ் இப்ப ஐசோசிட்ரேட் டீஹைட்ரோஜனேஸ் பாஸ்போரிலேட்டடா மாறிடும். அப்ப பாஸ்போரிலேட் ஆயிடுச்சுன்னா ஐசோசிட்ரேட் டீஹைட்ரோஜனேஸ் becomes inactive. ஐசோசிட்ரேட் டீஹைட்ரோஜனேஸ்ல அமினோ ஆசிட்ல ஒரு பாஸ்பேட் குரூப் அட்டாச் ஆயிடுச்சுன்னா அது இன்ஆக்டிவா மாறிடும். அதால சிட்ரிக் ஆசிட் சைக்கிள ப்ரொசீட் பண்ண முடியாது. ஆனா பாஸ்பேட்டேஸ் பாத்தீங்கன்னா it is going to remove the phosphate group from isocitrate dehydrogenase. அதுல இருந்து அந்த phosphate group remove பண்றது phosphatase. அப்ப என்ன ஆயிடும்? ICD அதாவது isocitrate dehydrogenase ஷார்ட்டா ICD will become active now and it can enter into citric acid cycle. அப்ப இப்ப protein kinase வந்து phosphorylate பண்ணி ICD inactivate பண்ண போதா இல்ல phosphatase அந்த phosphate group remove பண்ணிட்டு activate பண்ண போதா? இத யார் டிசைட் பண்ண போறாங்க? Some of the factors are going to decide this condition. What are the factors? Intermediates of citric acid cycle, intermediates of glycolysis, AMP, ADP இதெல்லாம் என்ன பண்ணும்னா டிசைட் பண்ணும். இப்ப intermediates of citric acid cycle glycolysisலாம் என்னென்ன இருக்கும்? எல்லா intermediates இருக்கு. நிறைய NADH-ஐயும் FADH-ஐயும் ஃபார்ம் பண்ணி ரெடியா வச்சிருக்கு. இந்த இன்டர்மீடியட்ஸ் எல்லாம் என்ன பண்ணும்னா எதை டார்கெட் பண்ணும்? AMP இருக்கு, ADP இருக்கு, ஆனா ATP இல்ல. So AMP, ADP லாம் பாஸ்போரிலேட் ஆகி ATP மாறணும் அப்படின்னா அது கண்டிப்பா சிட்ரிக் ஆசிட் சைக்கிள்குள்ளார நுழைஞ்சு அதுக்கப்புறம் எலக்ட்ரான் டிரான்ஸ்போர்ட் செயினுக்கு போனாதான் அது ATP மாற முடியும். அப்ப இந்த இன்டர்மீடியட்ஸ் எல்லாம் அதிகமா இருக்குன்னா என்ன அர்த்தம்? there is some lack of energy. எனர்ஜி இல்லைன்னா அப்ப அது எந்த சைக்கிளை சூஸ் பண்ணும்? it will be choosing citric acid cycle. So citric acid cycle சூஸ் பண்ணோம் அப்படின்னா அப்ப அதை என்ன பண்ணி ஆகணும்? யாரை ஆக்டிவேட் பண்ணணும்? புரோட்டீன் கைனேஸ ஆக்டிவேட் பண்ணிடுச்சுன்னா இம்மீடியட்டா அது ஐசிடியை போய் பாஸ்போரிலேட் பண்ணும், TCA சைக்கிள அரெஸ்ட் பண்ணிடும். பாஸ்பேட்டேஸ் ஆக்டிவேட் பண்ணுச்சுன்னா ஐசோசிட்ரேட் டீஹைட்ரோஜனேஸ் டீபாஸ்போரிலேட் ஆகும். அப்ப என்ன ஆயிடும்? ICD becomes active and it can enter into citric acid cycle. But now tell me the intermediates of citric acid cycle, glycolysis, AMP, ADP, all will be activating one enzyme. What is the name of the enzyme? Protein kinase, phosphatase. So the intermediates of citric acid cycle, glycolysis, AMP, ADP, all will be activating one enzyme. What is the name of the enzyme? Okay, I'm repeating the regulation. Intermediates of citric acid cycle, glycolysis, AMP, ADP will be activating phosphatase so that the phosphate group from ICD will be removed and ICD will be actively participating in citric acid cycle, and it is helpful for the conversion of isocitrate to alpha ketoglutarate, and alpha ketoglutarate will be entering into citric acid cycle and finally forms NADH and FADH2, which will be entering into oxidative phosphorylation or electron transport chain to form ATP. At the same time, these intermediates AMP and ADP will be inactivating protein kinase. This symbol represents inhibition, so that the protein kinase can also be inactivated. So inhibit பண்ணிடுச்சுனா protein kinase ஆல ICD inactivate பண்ண முடியாது. At the same time, these intermediates will also go and inhibit another enzyme called isocitrate lyase. Isocitrate lyase at the same time it will be inhibiting. Every protein kinase inhibit பண்ணதோ அதே மாதிரி isocitrate lyase inhibit பண்ணிடுச்சுனா glyoxylate cycle will not be proceeding. Only the phosphatase will be activated, so that ICD will become activated and TCA cycle will be continuing. And if these intermediates are not going to be present, all AMP and ADP are converted to ATP அப்படிங்கும்போது this protein kinase will be activated, and protein kinase will in turn inactivate ICD, so that TCA cycle will not occur. In turn, isocitrate lyase வந்து activate ஆகி glyoxylate cycle proceed பண்ணி it can form glucose, amino acids and nucleotides. So that's all about glyoxylate cycle. And once again repeating glyoxylate cycle. Glyoxylate cycle occurs in lower plants. Microorganisms such as E. coli, fungi, all will be carrying out glyoxylate cycle. The higher plants will not be carrying out glycolysis, because those plants which acquire the ability to carry out photosynthesis will not be carrying out glyoxylate cycle, and vertebrates do not possess the enzymes for glyoxylate cycle. And the important enzymes of glyoxylate cycle are isocitrate lyase and malate synthase, and these are the key enzymes of glyoxylate cycle. And glyoxylate cycle needs two acetyl-CoA molecules for the formation of glucose, and 4 different organelles are going to be involved in the formation of carbohydrates. The first one is lipid body, in which triglycerides are converted to fatty acids, and those fatty acids will be entering into glyoxysomes, and fatty acids are oxidized to form acetyl-CoA, and acetyl-CoA forms citrate along with oxaloacetate, and citrate to isocitrate, and isocitrate is going to be lysed into glyoxylate and succinate. The succinate which is formed with the glyoxysome will be entering into mitochondria for its further conversion to malate, and the formed glyoxylate combines with one more acetyl-CoA to form malate with the help of the enzyme malate synthase, and malate is oxidized to oxaloacetate, and the cycle will be continuing as such. And the formed succinate, every time the succinate will be formed, and this succinate will go out of the glyoxysome and enters into cytosol, and through cytosol it will be entering into the mitochondria, where it is oxidized to fumarate and fumarate to malate, and malate will go out of the mitochondrial membrane to form oxaloacetate using cytosolic malate dehydrogenase, and where the reversal pathway of glycolysis called gluconeogenesis occurs in the cytosol to form finally sucrose. So 4 different organelles are going to be involved in the formation of glyoxylate cycle, and ultimately we have obtained sugar molecules. And coming to the regulation part of glyoxylate cycle and citric acid cycle, isocitrate is considered to be the central molecule of both the cycles. Isocitrate is going to be converted to alpha ketoglutarate and enters into citric acid cycle with the help of ICD called isocitrate dehydrogenase. At the same time, this isocitrate can be converted to succinate and glyoxylate with the help of the enzyme isocitrate lyase. Any one cycle should happen at a time, either TCA cycle or glyoxylate cycle. And the isocitrate dehydrogenase, if it gets phosphorylated by protein kinase, it becomes inactive. If it is dephosphorylated by phosphatase, it becomes active. The phosphatase gets activated by the intermediates of citric acid cycle, glycolysis, AMP and ADP. The phosphatase, if it gets activated, it will be removing the phosphate group from ICD, so that TCA cycle will be continuing. And at the same time, it will be inhibiting protein kinase that is going to phosphorylate this enzyme. At the same time, it will also be inhibiting isocitrate lyase, so that glyoxylate cycle will not be proceeding simultaneously. And if ATP molecules are reduced in enough amounts, this isocitrate lyase is going to be activated, and glyoxylate cycle will be continuing. An oxaloacetate will be formed, and finally glucose, amino acids and nucleotides can be formed in sufficient amounts with the help of glyoxylate cycle in lower plants. So that's all about glyoxylate cycle. Whether this glyoxylate cycle occurs in higher plants?
No, ma'am.
Okay. So what is the end product of photosynthesis? What is the end product of photosynthesis?
Carbohydrates.
Carbohydrates, ma'am.
Very good. Which is released as the waste product of photosynthesis?
Oxygen.
Good. Okay. Vertebrates. Whether vertebrates possess enzymes for glyoxylate cycle? Vertebrates possess enzymes for glyoxylate cycle? Yes or no?
No, ma'am.
Answer. Tell me the answer. Unmute and answer me.
No, ma'am.
Good.
It doesn't contain.
Okay. And microorganisms. Some of the microorganisms are having the ability to carry out glyoxylate cycle. Name any
Yes, ma'am.
microorganism. Name any microorganism that has the ability to carry out glyoxylate cycle.
Bacteria, fungi, E. coli.
Bacteria, fungi, protists.
Okay, good. Whether glyoxylate cycle and TCA cycle occur simultaneously?
No, ma'am.
How it is going to be regulated? There are two enzymes that are help in the regulation of glyoxylate
Protein
cycle.
kinase. Protein
And
kinase
phosphorylation.
Wait. I'll finish my question. There are two enzymes that participate in the regulation of glyoxylate cycle and citric acid cycle. What are they? What are they?
protein kinase and phosphatase.
Very good. Protein kinase and phosphatase. Again and again. Only few people are answering me. Okay. Protein kinase phosphorylates isocitrate dehydrogenase. So that isocitrate dehydrogenase becomes dash. Active or inactive?
Inactive.
Inactive, ma'am.
Very good, very good.